Okay, so it gives me pleasure to introduce myself. So until they burst in and kick me out of this, I'm going to try to speak here as far as I can get with this talk. We had wonderful mitochondria presentations here. What I will try to do is just fill in some gaps why I think mitochondria is still important for the oocyte, and then go over the proposed treatments for oocyte quality for potentially mitochondrial aging. We've already heard about how bizarre of an organelle mitochondria is, which is a double-strand circular DNA molecule. And as a matter of fact, there's multi-copies of that in each mitochondria. And when they talk about mitochondria numbers, oftentimes these articles refer to DNA copy number, which is not necessarily mitochondria number, because there are multiple copies of that. Again, the fact that was emphasized is that even though we talk about mitochondrial genes, a lot more are on nuclear DNA that control mitochondrial function. So that the nuclear genome has a more significant role in mitochondrial function. But there are also interesting things about mitochondria that respond to needs of the cell. So if the cell needs higher energy, then mitochondria may grow and divide. On a low energy state, may be destroyed or become inactive. And it was mentioned fission and fusion. Mitochondria can actually fuse together, correct each other. But mitochondria's role is not limited to just energy. It does a lot of other things in the cell, and that's why I think that what David is saying is true. At the same saying that mitochondria is not necessarily important for energy, but it's important for many other cell functions, such as redox functions, beyond redox functions, oxygen sensing, fatty acid oxidation, calcium hemostasis, cell signaling, program death. So it wears many different hats in the cell. Now, there is a prevailing theory of mitochondrial theory of aging, and we always talk about the reactive oxygen species. Normally, oxygen is reduced to water through redox reactions in mitochondria. However, there's a very small amount of leak of superoxide radical during this process, which in itself is not damaging, but it has to be go through a process with lipid peroxidation to become an oxygen radical. So that's what could be damaging to DNA or genome. So the theory is that you have a little bit of leak, and then you get these oxygen radicals, and these do further damage. As a result, your mitochondrial function is now further damaged, further compromised, so you produce a little bit more of these reactive oxygen species, so you got that vicious cycle. Eventually, your cell loses its energy functions, and signaling and aging happens. So spinning off from that, people have hypothesized that with aging, this might be happening in the oocyte. And again, a second hypothesis is that meiosis is highly dependent on energy. Then therefore, with age, or mitochondrial energy functions are going down. B, because of that, now you cannot have a proper meiotic function. As a result, maybe this could explain age-related aneuploidy. So we've been, we've been going through lecture circuits with Dr. Keefe, and we did a debate on this a couple of years ago at FACOG. So I'm going to go over a few things. Well, let's see if we're going to end up like this. So let's look at the facts about mitochondria in the oocyte. The oocyte has a very large endowment of mitochondria or DNA copy, as high as 600,000, probably in several 100,000 on average. Morphologically, they seem to be inactive. Some have questioned that assessment, but they don't assume this active look until they're in the later embryonic stage. However, there's a lot of shuffling going on with mitochondria in the oocyte. The localization will change based on stage. For example, in the GV or M1 stage, it's perinuclear. Hypothesis, is it perinuclear because it wants to support meiotic activities or spindle formation or chromosomal segregation. Becomes more peripheral in M2. Again, becomes perinuclear at 2p stage. So it's not sitting there in a passive fashion. They talk about transient microzonation where mitochondria concentrates around the areas of need in a cell as well. So these are active players, perhaps shuffling around, depending on the cell's needs. Now, there's a slide. This study is sort of the debate between Democrats and Republicans. And Republicans look at the economy and say, "Oh, we're sinking the make America great again." And Democrats will look at it. We've got the top economy in the world. So this study is like that. Depending on how you look at it, it supports or refutes the important role of mitochondria. So this shows you how many copies of mitochondrial DNA you have in different stages of germ cell development. So it's at its lowest level at primordial germ cells, but as you develop into primordial follicle and a mature oocyte, the numbers peak. After fertilization, as these mitochondria are split between elastomeres, you get fewer and fewer. As a matter of fact, one of the reasons that's cited for large endowment mitochondria is that so that there will be enough for each embryonic cell in the end when embryo develops. So there will be sufficient number of mitochondria. So in this series of experiments, what they did was to reduce number of mitochondria and see how it affected subsequent implantation and embryo development. So not until you reduce the threshold to about 50,000 from about 200,000 that you would see effects. What happens with these embryos is that there's eventually spontaneous abortion. You have to reduce it to about 20,000, in which case the embryos implant, but there's no organogenesis. So that's severe mitochondrial failure. So this tells us that there's a lot of redundance, but you still need certain critical number of mitochondria to get healthy embryos or healthy oocytes. Now, in the natural state, and this is a porcine experiment, there are certain oocytes that have lesser mitochondrial function. So this is detected by brilliant crescent blue dye method. So those that stain with that has a better mitochondrial function. So in these experiments, they separated the sibling oocytes based on that. So those that have a better mitochondrial function, they have higher fertilization rate both by IVF and ICSI compared to those BCB negative. And if you take these BCB negative, so weak mitochondria type oocytes, and you inject them with BCB positive oocytes cytoplasm, you actually improve their fertilization. So it's kind of the cytoplasmic transfer concept. But if you do sham injection, their fertilizations are still lower. So supplementation of mitochondria or cytoplasm from more mitochondrially mature sibling oocytes actually corrects for this deficiency, suggesting that mitochondria has a role in fertilization. Dr. Keefe's theory of telomere dysfunction actually also ties into this because Dr. Keefe's laboratory has showed that mitochondrial dysfunction actually could lead to telomere attrition and genomic instability. So this could have a relationship to telomere dysfunction theory. Now, Dr. Keefe and I, had these discussions in the past, and one of the studies that suggested that in oocytes there may be actually alternative pathways. You do not necessarily need OXPHOX, and that's one of the pathways is called organism salvage pathway. So according to this paper, mitochondrial function actually is turned off from GV to M2 stage. So you're seeing that mitochondrial function is significant in GV oocyte, but it's turned off in M2 oocyte. But we have to, again, pay attention to that there's a significant role in the GV stage. And So it could still be relevant to oocyte quality, even if we take this bovine model as valid for all species. What about the clinical evidence that oocyte mitochondria might matter? A couple of correlative studies. This study looked at oocyte mitochondrial DNA copy in ovarian insufficiency patients. Here you have the control group. This group is patients, sort of PCO-type patients. And these are discarded oocytes, obviously, from IVF cycles of these patients, but they're severely reduced mitochondrial DNA copy number. And same thing for IVF cycles where there is unexplained fertilization failure. These are the copy numbers for those patients versus normal. So again, these are discarded GV oocytes. You can argue that may not represent the physiological condition, but at least some clinical studies suggested association. Now, in terms of mitochondrial diseases and fertility, there are some interesting clinical syndromes. The syndrome that is caused by Pathology deficiency, that's a mitochondrial DNA polymerase, and this is encoded by a nuclear mitochondrial gene, by the way, results in various neurological findings, but also premature menopause. So if you look at the pedigree here, the circles are obviously females who are affected, the dark, and the number is the age at menopause. So there's a persistent early menopause in these patients, age 32, 35, 28. And now also another study that which you can interpret both ways is this letter in New England Journal of Medicine, which was published to determine how many children would benefit from mitochondrial treatments. And so they arrived at 232 annually. But what they did was they compared the fecundity of these, live birth rate of these patients with mitochondrial disease to general population. Now, the problem with this study was that they used all patients who came to Newcastle, because that's the center that does all these treatments, and compared them to statistics from the whole of the country. So they're very different patient populations. Even though the overall there is no difference, when you look at subgroups, I see something different. I personally think that in these mitochondrial patients with mitochondrial diseases, there's also an added factor of age. If you look at the age group 35 to 39, where we see the most striking differences, that you actually have difference approaching significance. So I'm not ready to write off the possibility that in aging group of patients with mitochondrial dysfunction, there could be some decline in fertility. And finally, recent GWAS studies also identified mitochondrial dysfunctions, one of the possible determinants at age at menopause. So I think there is indirect evidence in humans that there may be some role of mitochondria in oocyte quality, but there's a lot of discussion around that. And since mitochondria plays multiple roles, it doesn't have to be through energy. It could be through various cellular functions. But we're still missing direct evidence that mitochondria function declines with age in human oocytes. So what we've been doing is trying to look at mitochondrial DNA damage. This is one of my past PhD students, Bahar, now back to Turkey, looked at some of the genes related to DNA repair in mitochondria in mice. So TFAM is a transcription factor involved in replication of mitochondrial DNA, but it also binds to DNA damage sites there. So the dark bars represent mitochondria from oocytes, single oocytes from young mice, and the lighter bars, the green bars, older mice. So these are single oocytes. So what we are seeing is that in the mouse model, there is increased expression of TFAM in older mouse oocytes. TDP1, which is involved in single strand break repair, there's an increase in that, and PARP1 as well. Whereas TERF1, which is the double-strand DNA repair, involved in double-strand break repair, is not increased. So there seems to be some evidence, at least in mouse, that there may be increased DNA damage and in response repair. Then we decided to study this in human tissue. What we are doing here is obtaining human ovarian tissue from organ donors. These could be various ages from prepubertal to advanced ages. With laser capture, we're capturing human primordial follicle oocytes, and then they are then subjected to library preparation and RNA sequencing to determine how genes related to mitochondria could be affected by age. So this is very preliminary data yet. In humans, we're not seeing any significant change yet with TFAM. However, NEAL1, there seems to be increased response in the older primordial follicles, suggesting there may be a damage response on the single-strand break side. Interestingly, in humans, again, preliminary, the double-strand repair, just like what we found in genomic side, nuclear genomic side, there may be some decline. So this preliminary data suggests that there's maybe something going on with mitochondrial DNA repair mechanisms as well with age. So with that, we've seen throughout multiple presentations of how to correct mitochondrial disorders, but I want to quickly just talk about autologous mitochondria injections, a highly debated point. These autologous mitochondria injections are based on some research suggesting that there could be cells in the ovarian cortex that could renew themselves and differentiate into oocytes in adult life. However, this is being refuted also by many groups and became a significant debate whether these cells exist or not. Recently, several groups obtained the antibody from, at least one of them from Dr. Tilly, to duplicate some of these findings. So the DDX antibody, this recognizes domain, which is supposed to be an intracellular domain, that is used to sort these cells and used for clinical purposes. So when they use this antibody to sort these cells and subsequently subjected them to PCR, they found interesting things. First of all, they found that they were able to pull various kind of cells with that. When they applied it, for example, mouse kidney, they could pull kidney cells and other cells. So it wasn't very specific. Secondly, when they analyzed the cells they pulled by PCR, they did not find germ cell markers in there. However, Dr. Tilly has written a response to that, saying that the antibody has improved and also their methods were not sensitive. And when they use immune precipitation, they could actually pull the right amount of material and they could actually detect it. So again, this argument goes back and forth. Nevertheless, a company called Ovasynes capitalized on this to use these cells as a source of fresh mitochondria in an autologous fashion. The idea that because they're renewing themselves, therefore they're renewing their mitochondria as well. So this was based on a couple of observations. First of all, the mitochondria from these what's called OPCs morphologically resembled to that of human oocyte. And they produce in vitro high levels of ATP compared to mesenchymal stem cells, ovarian somatic cells, embryonic stem cells, or induced pluripotent stem cells. So they have a good ATP potential. And also in preliminary unpublished experiments, they saw that these cells actually didn't contain some common mitochondrial deletions. With that, they came up with this augment treatment where ovarian biopsies are obtained from the same patient. These cells are isolated using the DDX antibody. And then eventually, during the time of ICSI, they're injected with the sperm in the oocyte to generate embryos to enhance oocyte quality. So I have to declare that I'm no longer a medical advisory board member for Ovascience, but I was at the time, and only those who were members could be involved in any of these work. So I propose to do this in Turkey. So with very valuable colleagues, we studied this, and I should acknowledge them, Dr. Baltacı, Sönmezer, Turan, Ünsal Baltacı, wife, and Aktuna and Moy. We looked at this validity of this treatment. This was actually a small study. We had 17 patients with at least 2 IVF failures with poor embryo quality. Of those, 2 were excluded because we didn't have prior IVF data. In this study, we compared post-treatment outcomes to their pre-treatment outcomes. So if we didn't have that, then we couldn't use them. So that left us with 15 patients undergoing ovarian stimulation. Of those, 2 canceled due to either poor embryo development or aneuploidy after PGS, nothing to transfer. 3 did oocyte pooling, so we didn't go ahead with them. So that left us with 10 patients undergoing embryo transfer. 4 had PGS and 6 did not have PGS. These are all frozen. This slide shows you the summary of the patients. Their ages range from 27 to 41. When you look across, you will see that some of them do have signs of diminished ovarian reserve, maybe AMH of 1.1 or elevated day 2 estradiol, but not all of them. But all had issues with embryo development, nonetheless. So this shows results from the embryology, embryological variables. The number of oocytes retrieved were similar, but of course there was a trend because these patients were now much older. Now that they have many times tried and failed in earlier years, and they were older now when they tried this treatment. But the biggest difference we saw was in fertilization rates. This was the comparison of their first failed cycle to post-augment cycle. There was no difference in number of 2PN embryos, but there was a borderline improvement in embryo grading. This table summarizes, again, these patients and number of pregnancies. So to summarize those, of those patients, 4 had PGS, 1 had a live birth, 1 ongoing pregnancy, 1 pregnancy loss, and 6 did not do PGS, and there was a pregnancy loss. So in this very small study, clinical pregnancy rate came to something like 40%, and live ongoing pregnancy rate about 20%. How did that, again, this was a study where we compared them to their past cycles, but how did that compare to success rates of other patients with similar characteristics? By the way, these are the patients who had conceptions. This is the live birth patient who had live birth. Some of the embryos prior to transfer, this is a representative embryo for some of the embryos prior to transfer, and post-augment treatment. So it's pre-augment treatment, post-autologous mitochondrial injection treatment. And this was the baby, first baby supposedly in Turkey born from this procedure. So in that clinic, in patients with similar age and number of IVF failures, one would expect a live birth rate of about 5%, and from the study it was about 20%. Again, with these small numbers, there's no significance there. How about the data from other centers? There were 2 other centers which did much larger number of cases with this. One is the TCART in Canada, Toronto, and FACOG clinic in Dubai. So they've done, and I can only present to you published data. They might have done more now. Altogether, maybe about 100 cycles. This is their patient population is a little different. They accepted patients who had as little as single IVF failure. but as many as 5 or 16. So if you go to ongoing clinical pregnancy rate and live birth rate per initiated cycle, for it's 26% per initiated cycle and 35% for embryo transfer from Canada, and from United Arab Emirates it's 18 and 32%. However, when you break them down to ages, and this shows success rates or the pregnancies across age groups, all pregnancies from Canada are under age 40, and there's only one pregnancy above age 40 from Dubai. As a matter of fact, at a recent meeting in Spain, Canadian group stated that they're not using this for over age 40. So it seems that this technique is not associated with any pregnancies over age 40 currently. The FACOG group did another small trial within that study. They took these oocytes from 25 patients, and after morphogenic analysis, they decided to put some of them to ICSI, some of them into Augment. Again, this is not randomized, and majority of the oocytes they put through were for Augment. And in the end, from the ICSI group, they had 2 embryo transfers, no pregnancies. From the Augment group, 14, they had 8 pregnancies. 9 of them, they didn't transfer at all. They were really bad. So again, this is not a blinded or randomized approach, and they were kind of choosing which one would go to Augment versus ICSI. But they suggested that the ones that had Augment had a better chance of reaching pregnancy based on this. I think to summarize all this, initial non-randomized studies suggest some improvement in fertilization, embryo quality, and possibly pregnancy rates under age 40 with OPC-derived mitochondria injection. However, specificity of these improvements cannot be proven from the current data. Whether this is the injected mitochondria or the injection itself, or you cannot determine from these studies. There will be a need for further prospective randomized studies before we can consider it effective and safe. I think there's a lot more future work to be done. First of all, we need comparative data on mitochondrial health. We don't know if the mitochondrial status of these cells going in the oocyte is any better than the mitochondrial health status of the oocyte that's receiving it. There's not that data. There's no randomized blinded study, either crossover or randomization different groups. However, the company does have a registry of children born from so-called Augment, and that would need to be seen. So I think whichever kind of mitochondrial treatments we are doing, either for mitochondrial diseases or trying to improve egg quality, we need to follow certain kind of oversight. I think we need the data safety boards and public transparency. By the way, these are recommendations again from UK, HIFA. While approving these treatments for mitochondrial disease, you need IRB oversight, you need long-term follow-up, you need very strict case selection. And if you're doing this, perhaps we could more responsibly consider some of these treatments. So with that, again, I'd like to thank you for staying for the latest session and listening to my talk. Thank you again.